Hi, everybody. We're going to get started in just a couple minutes. Could someone give me a quick sound check? If you can hear me, could you just type in yes into the chat? All right. That's good news. Thank you very much. All right. Thank you. All right. I have a series of yeses saying that you can hear me. We'll get started exactly at 1 o'clock Central Time. Oh, no, sorry, 1 o'clock Eastern Time, 12 o'clock Central Time, 11 o'clock Mountain Time, 10 o'clock Pacific Time. Is that what they are in California? Okay, is anybody else having a low volume issue? I've got my mic all the way up. I can adjust it a little bit more here maybe. Okay, so how is the volume now? And seeing that there's a lag, it'll take a couple seconds till I hear from anybody. Okay, great. I have better on the volume. So let's get started. It is 1 o'clock. I am Valerie May. I teach at the Woodstock Academy in Woodstock, Connecticut, and I'm here to go through Unit 6. For the AP Biology Review, this is on gene expression and regulation. So we are packed full of information. And for some reason, it's not going to full screen. There we go. Just as I say it, there it goes. All right, so first thing is thank you to all of the moderators of today's chat. I will try to keep an eye on the chat, but it's kind of hard to, because there's a lag, it's hard to be on the topic that I'm currently on and looking back at the chat. But our moderators will do a great job of answering all of your questions. And you can see we've got people from all over the country that are helping out today. So as with the other sessions, there are links below the video, there's links to the slideshow. I'm going to talk a little bit about some of the things I put into the slideshow that you can look back at later. And there's also a link to the schedule of the remaining reviews, so there's still quite a few reviews left to go because we got some time. We still have some time before that AP exam starts. There's also a question submission form for the remaining review sessions. If you submit your questions ahead of time, that would be great because we need some time to get everything together so that we can address all of your questions. So how to use this video. So same with all the other videos that you've seen so far. It's basically we're reviewing the big concepts. We have a ton to go through with Unit 6, and I'm going to try to hit everything we possibly can. But we got to get through it in a short amount of time. So to think about how the essential knowledges from this unit might be incorporated into this year's exam, I have a series of past FRQs at the end. And I've recorded my explanation, so you could do the FRQ and then hear my explanation and see what I wrote after each one. I'm not going to go through all of those today because we just don't have time, but there's recorded explanations for each of the videos. And remember, the link to the slide is at the bottom of below the video. And this is definitely to support, not replace, any review material that you already have. So your teacher has probably given you a lot of materials already and given you access to things. This is really to supplement that. So this is going to be me flying down our genes as fast as an RNA polymerase. But I'm going to go fast, but I put a lot of information down here. Basically, all of the essential knowledges that apply to the slide that we're going to be on are in the speaker notes, so when you go back to the presentation, you can take a look at those. All right, so let's get going. So what is gene expression? When we say gene expression, we're basically talking about how DNA is the instructions for making RNA. So DNA is not made into RNA, but it's the instructions for making RNA, and we call that transcription. And then RNA is used by those ribosomes during translation to make proteins. So that's what we mean when we say gene expression. We call this essential dogma, DNA, RNA, protein. And we really go all the way to trait. So DNA, so we've got some DNA up here, and we're going to go through transcription and translation. That gives us proteins. In this particular case, it's an enzyme. And that enzyme doing its job results in that purple pigment, and we've got a purple flower. And we'll also talk about mutations as well, and how that affects this process of gene expression. So we're going to go over gene expression. There's a lot, so let's just get going. All right, so topic 6.1, RNA and DNA structure. So there's some things that you need to know. Basically, DNA in eukaryotes and prokaryotes are organized into these things called chromosomes. In eukaryotes, there's typically multiple chromosomes. In sexually reproducing organisms, they occur in pairs. You might remember that from the genetics, the homologous pairs. And those chromosomes, the DNA tends to be linear in those chromosomes. So there's a beginning and an end. Whereas in prokaryotes, bacteria, they tend to have a single chromosome. That chromosome is circular, so there's no beginning or end to the DNA molecule. They do have these small plasmids, and we'll talk about those plasmids in a little bit. I'm going to talk about those plasmids right now. So those plasmids are actually used. So I'm jumping to topic 6.8 because that's biotechnology, and I'm going to sprinkle that through as we go through the different topics. So we can use those small circular pieces of DNA, those plasmids, in something called genetic engineering. So the idea is you could take some DNA from a eukaryotic organism, take a chunk of it. You can then put it into a plasmid that's using those restriction enzymes and DNA ligase. So we make what's called a recombinant plasmid. And then you do a bacterial transformation where you get that plasmid to go into a bacteria cell. Some of you might remember this from the PGLO lab My little graphic isn't doing it what it's quite supposed to do. There we go. There's the green fluorescent protein. And so remember, there is a recombinant plasmid, and that plasmid had the GFP gene on it, but the GFP gene doesn't glow green. It's the protein that the bacteria can make that gives you those glowing green bacteria cells. So there is a little bit of a deviation sometimes from that central dogma, and that is we have these things called RNA viruses or retroviruses. So not all RNA viruses are retroviruses, but the idea here is instead of the genetic information that's being passed from one virus to the next being DNA, the genetic information is RNA. Sometimes that RNA is used directly to make proteins, and then other times that RNA is used by reverse transcriptase, and that's made into DNA, and then the DNA is transcribed into RNA, which is then translated into viral proteins. So like COVID, the coronavirus is an RNA virus. Many viruses are retroviruses, HIV, influenza, those are all retroviruses, and they have RNA as their genetic information. So the question is, are the plasmids just rings of recombinant DNA? So I'll just quickly go back. The plasmids are actually naturally occurring in the bacteria, so they don't have to be recombinant DNA. And I'll talk about how bacteria use those to increase their genetic variation at the end. So what makes it a recombinant plasmid is that you've done this process, this laboratory process, where you've cut it with restriction enzymes, used DNA ligase to insert DNA that wouldn't normally be found in that plasmid. All right, so I do want to draw your attention down here to this video. There was a lot that went through on DNA and RNA structure in Unit 1. So I'm just kind of picking up some highlights that are part of Unit 6. So there's base pairing in DNA. So adenine base pairs with thymine. Cytosine base pairs with guanine. When we're talking about RNA, RNA has a uracil nitrogenous base instead, and that would base pair with an adenine. So there are these things called purines and pyrimidines. Memorizing which are which is kind of one of those things that you got to decide how much space in your brain do you want that to take up. It is in the course description, so it is fair game. I would basically remember that a purine always base pairs with a pyrimidine, and that's because the DNA has this parallel nature to it, and so we've got the purines, which are the double rings, and the pyrimidines, which are the single rings, and so a purine is always going to base pair with a pyrimidine to keep it that parallel structure. So our moderators are doing a great job in the chat. I just can't keep track of it while I'm going through. So thank you so much for doing that. So again, there's a lot more of information on the Unit 1 review, and I've given you the link there. All right, so if you notice, there are some negative charges on these phosphates right here. So here's a negative charge, there's a negative charge. These phosphates of DNA, of the nucleotides, we actually can use those phosphates to do some more biotechnology. So we got this thing called DNA electrophoresis. There also could be RNA electrophoresis, and the idea of that is we're going to separate the molecules based on their size. And just trying to catch up with the chat for a little bit. Yep, loving everything that the moderators are saying. So thanks a lot. So the way DNA electrophoresis works is DNA has a negative charge, and so you have this agarose gel, and you set up some wells, little impressions that you can load your sample into. You put those by the negative, and then you go, you have a positive, so you have this power source, this electricity going through, creating an electrical field. And because the DNA has that negative charge, it's going to be attracted towards the positive, repelled from the negative, and so it's going to migrate through the gel. But then it's going to be sorted by its length, because the idea is shorter fragments move faster through that gel. And so you load it into the wells, you turn the power on, and then those largest fragments move very slowly through the gel, and those smallest fragments move very quickly through the gel. And so the whole idea is that electrophoresis separates DNA and RNA by the length using the electricity. And I will go back. We're going to come back to retroviruses. This isn't the last time that we've talked about it. So we will come back to those again. Okay, so topic 6.2, DNA replication. Again, we got a lot to get through. We got a lot more topics to go. Everything is being recorded, so you can go back and watch certain sections again. And remember that I have all of the essential knowledges down in the speaker notes down there. So topic 6.2, we're supposed to describe the mechanisms by which genetic information is copied for the transmission between generations. So DNA is copied by this process called DNA replication. And one of the things to know about DNA replication is, again, I'm going to come back to retroviruses. So definitely going to come back to those. They show up in a later topic. So DNA replication is done by this process called semi-conservative replication. And all that means is that DNA has two strands, and each of those strands are going to act as a template strand for the synthesis of a new strand. And because G matches with C and A matches with T, that complementary base pairing, you're going to end, even though the two template strands have different sequences, you're going to end up with two identical molecules after DNA replication takes place. Another important thing to remember is that DNA is synthesized in a 5' to 3' direction. And I will talk more about that when we talk about the different enzymes involved in the process. But you can see here on the green, so this green right here, this is the newly synthesized strand. And so DNA polymerase is always going to be adding nucleotides to that 3' end. But because our DNA is anti-parallel, 5', 3' on one strand, the opposite strand goes 3', 5' in the opposite direction, we're going to have a little bit of a complication on how the two strands are replicated. All right, so topic, so we're still here. So DNA is synthesized but in a 5', 3' direction, and what you basically want to remember is that DNA polymerase can only add a nucleotide to the 3' hydroxyl group of the strand that it's making. So you can see right here, this is the new strand that's being made. And so here is that hydroxyl group. And so this new nucleotide has to be added to that hydroxyl group. A new nucleotide couldn't be added to a phosphate or a 5' end. And that's one of the ways that I remember 5' end has the phosphate. So use your alliterations, FF, even though it's PH, you got it. Okay, so we got a lot of enzymes involved in DNA replication. There's a lot more than you need to know, but I'm going to go through the ones that are listed in the course description. So the first two are helicase and topsomerase. So DNA is a double helix. The two strands are all connected by hydrogen bonds, and basically the enzymes can't get to the template when it's in that double helix. So we have two enzymes that are involved in opening up the two strands so that we have two templates that DNA polymerase can use to synthesize the new strand. And those two enzymes are called topsomerase and helicase. So helicase has the job, there's a great video that I linked at the end of replication. We're not going to take the time to watch it during this session, but it's available in the slides for you to go back and look at later. And in that animation, it's a blue helicase that's spinning really, really fast, and as it spins, it unwinds and unzips the DNA. Remember, unzipping just means it's breaking the hydrogen bonds that are connecting the nitrogenous bases. So it's opening it up into single-stranded DNA. The problem with that is that it creates some twisting tension, and so topsomerase just has the job to alleviate some of that twisting. It makes little nicks, and then they connect again. So that if you've ever tried to take two strings that are in two threads that are in a string and pull them apart, you end up with this big knot at the bottom. Kind of the same concept. So topsomerase just alleviates some of that twisting tension. And again, this information is listed down in the speaker notes. This diagram, all I'm showing you is the difference between that circular versus linear DNA. So up at the top, we have prokaryotic DNA, circular DNA. And so the helicase would, you basically get two helicases, which result in two replicating forks. One helicase goes in one direction, the other helicase goes in the other direction until replication is done. In eukaryotes, we've got linear DNA, and so we have multiple origins of replication. And so pairs of helicases will bind to each one. They open up those replicating forks, and then the rest of the magic that we're going to talk about happens right here. All right, so DNA polymerase, I like to call it a diva. DNA polymerase can only work under certain conditions. It must have a 3' hydroxyl group to add to. If it doesn't have a 3' hydroxyl group, DNA polymerase can't just hop onto single-stranded DNA and start copying, at least the DNA polymerase that's doing this part of DNA replication. And so what there has to be is there has to be a primer, and that is an RNA primer that's put down by RNA primase. So RNA primase, as soon as that single-stranded DNA is available, RNA primase puts down a little RNA primer, little RNA nucleotides, and then that provides the 3' end for DNA polymerase to add to. So what does DNA polymerase do? DNA polymerase adds nucleotides to the 3' end of the strand that it is making. And it does that by, if there's an adenine on the template strand, it puts a thymine into the new strand. Cytosine, guanine, guanine, cytosine, thymine, adenine. So it uses that complementary base pairing. Basically, what it does is it makes the sugar phosphate bond that locks that nucleotide into the polymer. And so it's doing that dehydration synthesis that puts the monomers together into a polymer that we've heard about in Unit 1. Okay, so that brings us to this whole idea of leading and lagging. So there is a leading strand and there is a lagging strand. So there's actually two DNA polymerases working at each replicating fork. So if we go back here for a second, so there's actually two replicating forks right here. There's two helicases. And so all of these enzymes that I'm talking about, there's a set of them at each replicating fork. So every replicating fork has its set of DNA replication enzymes. We sometimes call that a replicosome. So leading and lagging strand. So remember that DNA polymerase can only add to the 3' end. It needs that hydroxyl group. Well, one strand is in the correct orientation, so that there's always a 3' end for DNA polymerase to add to. And as DNA helicase unwinds and unzips and continues to feed that single-stranded DNA, there's always a hydroxyl group for DNA polymerase to add to. The other strand, because there's an anti-parallel nature of the DNA, the other strand, DNA replication, has to happen in the opposite orientation. The 3' end is always here. And the problem is this DNA polymerase is going to go, and then it's going to bump into what it's already made. It's going to run out of single-stranded DNA. And so then, I think this diagram shows it a little bit better. So this was kind of in sequence in which it was made. This fragment was made, then this fragment was made, then this fragment was made, and now this fragment is being made. So this is called the lagging strand, and that's because DNA polymerase can only add to the 3' end, and this particular strand is running in the opposite direction of the lagging strand. So this is actually pretty complicated, but the video that I have coming up in a couple slides would be excellent to go back and read over again. The big point is DNA polymerase can only add nucleotides to the hydroxyl group on the 3' end of what it's making. So it needs that primer, and then it's going to extend that primer. On the lagging strand, a primer has to be put down for each of these fragments, which is called an Okazaki fragment. There are some other things where there's other enzymes that come in and replace the primer, but then we need this enzyme called DNA ligase to connect the DNA fragments. Again, this is pretty complicated. We got a lot more to go through. This right here is an excellent, excellent video that reviews everything that I just said. And that's available in the slides that you'll be able to access after this session. Okay. So this brings us to a biotechnology topic. This idea of how replication works was used to come up with this process called the polymerase chain reaction. The idea with the polymerase chain reaction is it's to copy specific fragments of DNA. And back when we did DNA replication, the entire genome is being copied. The entire set of DNA inside the cell is being copied. And remember, that DNA replication thing only happens during S phase of the cell cycle. So if you have a cell that's not dividing, then DNA replication is not going to happen inside that cell. So that's something important to remember about DNA replication. So the way PCR works is you have a section of DNA that you want to copy for some reason. You use a series of primers. You call up the company. You tell them what sequences you want for your primers. You need to have 2 primers, one for the beginning of the one strand of the target sequence, the other for the beginning of the other strand, because remember, DNA polymerase can only add to the 3' end. And so basically what happens is you heat it up, you denature the 2 strands, the primers bind, it's called annealing, and then this special enzyme called Taq polymerase is going to start adding nucleotides to the 3' end. It's a special type of DNA polymerase, and you're copying the 2 template strands. And here's another model, and so after one cycle, you have copied, you have 2 copies of your target sequence. They're not quite the right length yet, but eventually we'll get there to ones that have the right length. And then you keep repeating this cycle 25 to 35 times, and every DNA molecule that's been made is now acting as the template to make more DNA molecules, and eventually you end up with millions of copies of a very specific sequence of DNA. So where this might be used, and then you have to do DNA electrophoresis to actually see it. So you would end up with a band like this, which is the size between the 2 primers. And then how you might use this is you might use it in forensics. Again, not enough time to go through all the details. We still have a whole lot more to do. But the idea is like, here's your suspect. Here's the evidence. This is not the suspect does not have the same DNA as the evidence, but suspect 3 does. Yes, Taq polymerase can only work at high temperatures. Whew. All right, so topic 6.3, transcription and RNA processing. So remember, gene expression is really using the sections of DNA called genes to make some type of product, whether that's an RNA or most often a protein that's going to carry out some type of function in the cell. So here we go, transcription. Transcription, we're using a different enzyme this time. It's called RNA polymerase, and it's only going to copy a single template strand to make that RNA molecule. And while DNA polymerase needed all these other molecules to help it, RNA polymerase pretty much can do it on its own. We're going to find out it has transcription factors in eukaryotes, but pretty much can do it on its own. So I'll go through a little bit more of the details in a second, but you can see that there is one strand that's acting as the template strand. We sometimes call this the antisense strand. I'll give you a whole list of things that we call the template strand. And so pretty much it's like DNA polymerase, but instead of DNA nucleotides, we've got RNA nucleotides, and you can see right here that instead of a T being base paired with an A U is base paired with the A. Other than that, it's very similar. So it starts with RNA polymerase binding to a promoter sequence. We'll talk more about that in regulation of gene expression. So then RNA polymerase, it'll bind to that promoter sequence, and then it will be moving down. Again, there's a video for this that you can see, a narrated video. RNA polymerase will move down that template strand, and it will add nucleotides to synthesize what we're going to refer to as the pre-mRNA if we're talking about eukaryotes. Now, one thing about vocabulary. So the DNA strand that is being copied, so that would be this strand right here. This is the one that's actually being copied. We refer to that as the template strand. We also refer to that as the non-coding strand because it doesn't have the same code as the mRNA. It's complementary to the mRNA. The strand that's not being copied is called the coding strand because that essentially has the same sequence as the RNA molecule that's being made, except it has the coding strand would have T's, whereas the RNA would have U's. It's also sometimes referred to as the minus strand and the antisense strand. So all of these are listed in the course description. So it's kind of one of those small grain size pieces of information, but I would definitely remember that what's copied is called the template strand. And then the other strand of DNA is called the coding strand. Okay, just lost my track. All right, so remember RNA polymerase, it's just like DNA polymerase. It can only add to the 3' end of the strand that it is making. And then there's the base pairing rules. And then transcription is going to end when RNA polymerase reaches a terminator sequence. Now, this is not the same as the stop codon. The stop codon is actually upstream from this terminator sequence. The stop codon actually only is involved in translation. There's a sequence called the terminator sequence, and that's going to cause RNA polymerase to come off, and now you have your RNA molecule. And the DNA is going to basically zip right back up again and be unchanged by this process of transcription. All right, so if we were in a prokaryotic cell, we would actually be doing translation at the same time as transcription. There's no nucleus separating the two processes, and so as soon as that RNA is coming off the RNA polymerase in a prokaryotic cell, in a bacteria cell, there would be a ribosome translating it. But in eukaryotic cells, there's a couple more steps to go before we're done with this part. All right, so it's called RNA processing. RNA processing only occurs in eukaryotic cells, and it takes place inside the nucleus. So those are actually two really important pieces of information to know. So RNA processing only in the eukaryotic cells, and it occurs in the nucleus before the mRNA leaves the nucleus to go to the ribosome to be translated. And basically 3 things are going to happen. One thing that's going to happen is a poly A tail is going to be added. So there's an enzyme that adds a series of adenines to make a poly A tail. And that kind of protects the end of the mRNA from nucleases that are going to be chopping up the RNA. The longer the poly A tail, the longer the coding sequence is available for the ribosome. It can be translated multiple times. We also have a 5' cap, which is going to be added. So both of those help it, they're kind of like exit tickets to get out of the nucleus for the mRNA, but the 5' cap is also going to help the mRNA bind to the ribosome later in translation. And then we have these things called introns. So introns stand for intervening sequences. They are the sequences between the expressed sequences of the RNA. Expressed sequences are the exons. And so these introns need to be removed. And they are removed by a spliceosome. And yes, the spliceosome is made up of little things called SNRPs, which are just fun to say. So the spliceosome has the job of recognizing an intron, cutting it out, and then connecting the exons together. Remember, this is only done to the RNA. This is not done to the DNA. The DNA has been rezipped and is intact. It seems like it's this wasteful extra step, but it's something that helps increase some variation, and we'll see that a little bit more in a second. So now you have your spliced mRNA, and that can go off to do the translation step. So there's just more of the same thing. So then we come to this thing called alternative splicing. Alternative splicing, I didn't know about this when I was in college. It hadn't been discovered yet. And so that's one of the amazing things about molecular biology and biology in general is we're constantly learning new things. So alternative splicing is basically the idea that a particular gene, so here's our gene up here, here's our DNA, our gene, can be transcribed. We get the primary transcript. That primary transcript can then be spliced in alternative ways based on signals that the cell receives. And so in this case, we're seeing that the same primary transcript, the same pre-mRNA is being spliced 3 different ways, and it gives us 3 slightly different variations of the protein. One thing to remember is that all 3 of these proteins are coded for by the same gene. It used to be 1 gene, 1 protein, but we now know that 1 gene can actually code for multiple variations of the same protein. Just more ways to give us variation in the proteins that we have inside of our cells. Yeah, so the 5' cap is also, we're going to see it in a little bit when we get to translation. It's how the small subunit of the ribosome docks onto the mRNA. All right, so we now have a mRNA sequence that has its 5' cap and its poly A tail, and it is ready to go to the ribosome. So we are ready for translation. Check on our time. Okay. There are 3 types of RNA molecules involved in protein synthesis. We got the mRNA, the tRNA, and the rRNA. So the mRNA is a copy of the DNA. It contains the code that the ribosome is going to use to make a polypeptide. And we'll talk more about that in a moment. We have the tRNA. So this is an example of an RNA molecule that never gets translated. I'll give you some more details on the tRNA, but the job of the tRNA is to carry specific amino acids to the ribosome. So they're the ones that actually deliver the amino acids in the right order. And then there's the rRNA, which actually makes up the ribosome. So the ribosome is a series of RNA molecules wrapped around with some proteins. Siri's trying to help. There we go. And so rRNA would be in the ribosomes. So a little bit more on the tRNA. The tRNA has 2 really important regions. It has an anticodon down here. So remember, RNA is single-stranded. This is a single-stranded molecule. You could follow the backbone, but it does fold up. It uses base pairing to fold into a 3D shape that can fit into the ribosome. And then up here at the other end, so there's an anticodon down here, 3 base pairs sticking out, and then up here there's an amino acid binding site. The anticodon is going to specify which amino acid that particular tRNA is going to carry around. So there's enzymes that are involved in recognizing the anticodon and adding the correct amino acid to the tRNA. And that's really key for the whole translation process because the tRNAs need to be carrying the right amino acid. So a little bit more about the tRNA. That anticodon is going to be complementary to the mRNA codon, and this is how the correct amino acids are going to be brought to the ribosome and put into the right order based on the sequence of the mRNA. Okay, so translation. DNA is copied into RNA. We got this mRNA sequence, and we now want to make a polypeptide, which is going to fold into a protein. So we need to do this translation step. You'll notice here that the mRNA is broken up into groups of 3. So 3 nucleotides, 3 bases on the mRNA is called a codon. Remember, on the tRNA, it was an anticodon. So tRNAs have the anticodon, mRNAs have the codon. And they're going to code for the specific amino acids, or they're going to code for making the whole process stop. This is the codon chart. You should know how to read one of these. Basically, you start over here with the first letter, and then once you're in that particular row, you go to the column for the second letter, and then you find your third letter. And that's what that particular sequence on the mRNA codes for in terms of what amino acid should be put into the polypeptide. So translation does have 3 steps. We have initiation, elongation, and then there's a third step called termination, and we're going to quickly go through those 3 steps. But before we get into that, I just want to remind you that the DNA that you have, the version of the DNA sequence that you have, so is your genotype. So like we are sexually reproducing, plants are sexually reproducing, so we're diploid. So you get 2 copies of every gene. This particular organism would be heterozygous because they have a C versus an A. They have 2 different alleles. That's your genotype. Those alleles are then either going to cause nothing to happen, no enzyme at all. We'll talk about how that can happen with mutations. Or the allele will get transcribed and translated, and that makes a protein. And then proteins give us our trait, and that's the phenotype. So this particular learning objective is describe how the phenotype of an organism is determined by its genotype. So the DNA has been, the gene has been copied into RNA. That's our mRNA. And now we're going to use that mRNA to make a polypeptide. And then that polypeptide, if it folds into the right shape, is going to do what it's supposed to do, and it's going to give you a particular trait, phenotype. All right, so initiation. So we got our small subunit, and we've got our initiator tRNA carrying a MET, a methionine amino acid. That small subunit is going to find the 5' cap of the mRNA, and then it's going to kind of move along that mRNA until the anticodon of the initiator tRNA finds its complementary codon on the mRNA. So we got AUG. Once that process happens, the large subunit of the ribosome comes in, and now we've got all of our binding sites, and everything's going to happen to make our polypeptide. So now we're going to move on to elongation. So based on the sequence of codons on the mRNA, the correct tRNAs will base pair, and they will deliver the amino acids in the right order. So the large subunit has the job of making that peptide bond, taking the hydroxide off of the carboxyl group and the hydrogen off the amino group of the 2 amino acids, and then connecting them together to make that covalent peptide bond. Dehydration synthesis. Back to Unit 1. And this process is just going to keep going. So the ribosome keeps moving down 1 codon. The tRNA that doesn't have the polypeptide anymore is going to exit and go find another amino acid. New tRNA is going to come in, base pair its anticodon with the mRNA codon, delivering the next amino acid. A new peptide bond will form, and it just keeps going over and over and over again. So yes, the terms initiation, elongation, and termination apply both to transcription and translation. But again, the process is different because it's a different overall purpose. So like transcription, initiation is when RNA polymerase binds to the promoter, and then elongation is as RNA polymerase is traveling down making the sequence of nucleotides, and then termination is when it hits the terminator sequence and ends. All right, so how does translation terminate? Translation terminates because the ribosome is going to hit one of the 3 stop codons. There's really no tRNA for the stop codon, but there is a protein called a release factor that will bind. It will recognize those 3 stop codons, and this is going to cause everything to kind of fall apart. The 2 subunits of the ribosome are going to come apart. The polypeptide is going to come off that last tRNA. There could be other ribosomes right behind this one, so the same mRNA can be read by many ribosomes to make quite a bit of protein. And that's why that poly A tail is important, because meanwhile coming on that 3' end is enzymes that are chopping up this mRNA. So the poly A tail has that protective quality to it. All right. Whew. Here's the whole process. This would be an excellent picture to go back to on the slides. It takes you through the whole process of translation. So just there's a ton of stuff in this unit, a ton of stuff in this unit. And so one of the things to keep in mind is, where is that protein going to end up, and where is the ribosome going to be so that that protein can end up where it needs to go. So things that are going to be used inside the cell, so proteins that are going to be used in the mitochondria and the chloroplast, the nucleus, the cytosol, they're all going to be made on free-floating ribosomes, whereas proteins that are going to be secreted out of the cell or found in the plasma membrane or in other parts of the endomembrane system, such as the endoplasmic reticulum, the Golgi, or the lysosomes, those are going to be made on ribosomes that are bound to the rough ER. So a little review from Unit 2. Okay, so here's the whole thing. Oops, let me go back. Here's the whole thing. So DNA is copied into the primary transcript, and then that is spliced, and then the cap and the tail are added, and then that mature mRNA is used to make the protein, which is really it's just making a polypeptide, and that folds into that 3D shape. Unit 1. There are so many connections between the units, and that's what those AP FRQs are going to be. They're going to be asking you to really pull all of the different ideas from all the different units together. So back to that retrovirus idea. I promised we'd come back to the retroviruses. So how does the retrovirus fit into this whole thing? So this is actually for HIV. So you see up here we have HIV. It's bonding to a receptor. It's being taken in by endocytosis, receptor-mediated endocytosis. The virus then releases its RNA and uses its RNA, its reverse transcriptase, to copy its RNA into DNA. In HIV, the DNA is then inserted into the genome. And then something at some point in time will trigger that DNA to be transcribed, and then the RNA to be translated, and then new virus particles will assemble. So retroviruses do something a little differently. They have RNA, which is used to make DNA, and then the DNA is used to make RNA to make proteins. There are RNA viruses that are not retroviruses. And those RNA viruses, they have RNA, and that RNA is used directly to make proteins, and the RNA is copied to make more of those viruses. Okay. So I am definitely going to be going over an hour. I'm going to aim for an hour and 15 minutes. Remember, you got to go, but it is recorded and you can go back and you can listen to it. We've got 3 more topics to get through. So we've got 6.5, regulation of gene expression, and 6.6, gene expression and cell specialization. And I'm going to kind of mash these two together. Remember, there are the essential knowledges are in the speaker notes, so all of the concepts that I'm going over are in there for you to go back and look at later. So here's an example. We've got 2 different cells. Remember, every single cell in your body, for the most part, there are exceptions. Let's not worry about the exceptions. Every single cell in your body has the same DNA. So this gene to make alcohol dehydrogenase, it's found in a neuron. It's just not going to be expressed in the neuron. The gene for making the neurotransmitter is found in the liver cell. It's just not going to be expressed in the liver cell. So regulation of gene expression, which genes we turn on at what times, is how we get the specialized cells that we have inside our body, so cell differentiation. I like to refer to it as differential gene expression, and that just means that different times and or different cells. Genes are going to be expressed at different times under different conditions in a particular cell. They're going to be turned on, off, up, down. They also are going to be expressed. Some genes are not going to be expressed in this cell, but it is going to be expressed in that cell. So this is just an example of that. Okay, so prokaryotic gene expression. Regulation. We got this thing called an operon. An operon is found in bacteria, so this is really just focusing on bacteria right now. We have a promoter. Promoter sequence is always where RNA polymerase is supposed to bind to. It's kind of like the start of a gene. So every gene has its promoter, except here we got 3 genes. They all share a promoter. So this is a way to coordinate expression. In this operon, there's also something called an operator. So promoter, operator, and then the gene sequences, the coding sequences, these are all sequences of DNA. So this up here is all DNA. And we're talking about regulation of transcription for prokaryotes. That operator is a spot where this protein called a repressor can bind to. And if that repressor is binding to the operator, then RNA polymerase can't get past. So you want to think of the repressor as like a roadblock. We'll talk a little bit about the activator. An activator really just can help RNA polymerase bind better. It assists it. So you kind of have positive control with the activator and negative control with the repressor. So how does this work? We're going to use a lac operon as an example. And so when there's no lactose present, so this is called an inducible operon. So when there's no lactose present, the repressor protein binds to the operator, and RNA polymerase, it can't get past the operator. And so these 3 genes are not going to get transcribed. Now, these 3 genes here, these are genes that make proteins that help the cell to utilize lactose. So you want to kind of think about it like it doesn't make sense to make enzymes to waste all that energy for transcription and nucleotides for transcription and all the amino acids and energy for translation. You don't want to waste those if the thing the proteins are needed for isn't present. So when lactose is not present, these genes are off. They are not being expressed because it doesn't make sense for it would not be advantageous. It wouldn't be advantageous for the bacteria to make the proteins that these genes code for because the lactose isn't present. When lactose is present, so like this bacteria is hanging down in your large intestine and you just drank a whole lot of milk or just ate a whole lot of ice cream and some lactose got past your small intestine and didn't get digested, and now there's lactose down there in your large intestine, this bacteria is like, hey, look, there's lactose. I could use lactose. Well, right now it doesn't have the enzymes to do that yet, so it's got to turn on these genes and make the protein so that it can use the lactose. It actually uses an isomer of lactose called allolactose, but when there's plenty of allolactose around, it binds to that repressor, and the repressor is no longer binding to the operator. It changes its 3D shape so it can no longer bind. Now RNA polymerase has nothing blocking it, and so it can transcribe these 3 genes, the ribosome can translate the mRNA, and these proteins can get made, and the cell can utilize that lactose that is now available. So without getting too much into it, because it doesn't really specify in the course description, so allolactose is the inducer. This CAP site is where an activator would bind, and what happens is CAP is only going to, the activator is only going to be present when glucose is not around. So, yeah, I don't have that slide. But my advice to you is understand this. Repressor binding to the operator RNA polymerase can't get by. Can't transcribe, nothing to translate. Use any information that they give you in the prompt to help you figure out how a particular gene is being controlled. They give you lots of information, and part of it is you understanding your concepts. RNA polymerase transcribes genes into mRNA. mRNA is translated by ribosomes into a polypeptide. Understanding that repressors binding to the operator prevent transcription. Understanding that activators help transcription take place. You can then use any information that they give you for a particular scenario to figure out what they're asking you to figure out. All right, so eukaryotic gene expression. A little more complicated. There's more steps. It's more regulated. And that's probably because there's a lot more, it allows a lot more cell specialization. A bacteria cell pretty much just turns genes on or off, up or down in terms of environmental conditions. Eukaryotes tend to be multicellular, and so there's all these mechanisms of having differential gene expression to get cell specialization. So one level is up here at the very beginning. If the enzymes, RNA polymerase, can't get in there, if transcription factors, which we're going to talk about in a second, can't get in there, those genes can't be transcribed. So this is, it's kind of related to epigenetics. Again, that's a huge topic that we don't really have the time to get into the details, and we don't need to for the AP exam. So basically the idea is DNA is wrapped around these histones that make these, well, DNA is wrapped around these clusters of histones called nucleosomes. If the DNA is, the histones are all connected to each other, the DNA is like super tightly coiled up. And we actually call it heterochromatin. But then RNA polymerase can't get to it, and all those genes are off. It's like a file that's been zipped so that you can email it to somebody else because it's too big. So this gene right here would be considered off because it's part of this tightly coiled up DNA. And that's typically associated with the DNA being methylated. Again, small grain size, just giving it to you. Use the information that's given in the prompt. So the DNA being methylated and the histones being methylated cause that DNA to wrap up tight. No transcription can take place, so these genes are inactivated. Acetyl groups, they can open up the chromatin, and we call it euchromatin. And now, just as the diagram says, transcription factors can get in there, and these genes can be expressed. Doesn't mean they're necessarily going to be expressed, but here the gene cannot be expressed. Here it could be expressed if the other things are in place. All right, so what are those other things in place? So this is from a great activity by Biointeractive called Modeling the Regulatory Switches of the PIDX1 gene in the stickleback fish. Some of you might have done this activity. Here is the structure of a eukaryotic gene. There's typically a promoter. Remember, that's where RNA polymerase is going to bind. There's the coding region of the gene. That's what RNA polymerase is going to copy into that primary transcript. It says mRNA here, but remember, splicing has to happen first before you get to your mRNA. And then what we call upstream, so upstream, we have what we call enhancer sequences. Those enhancers are sequences of DNA that molecules called activators will recognize. All right. So somebody mentioned I saw Tata box come up in the chat as I briefly looked. So a Tata box is kind of what we call a conserved sequence that promoters tend to have. It's where RNA polymerase and a lot of general transcription factors will bind. And so, again, back in that transcription video, at the end of transcription, actually, I think they're in a couple slides. There's a transcription video and then there's a translation video. It'll show all of these general transcription factors binding to the promoter and recruiting and helping RNA polymerase get in there. And this right here, I tried to make it, didn't show it very well. But this is kind of a notation here saying that transcription is going to take place. So you sometimes see that in a model, and that says that transcription is going to take place. So once those transcription factors all assemble, this RNA polymerase can then go. There are some genes that have to have, a lot of genes. There's actually a lot of genes that have to have also activators binding to these upstream enhancers. And this is just another picture from that same activity, and it's showing that there's these 3 different enhancers so that the same gene can be expressed in multiple tissue types. And so what happens is those activators, when they're present inside the cell, they bind to the enhancer, and then the DNA kind of does this like folding thing, and the animation that comes up in a little bit shows this great. And it's really a combination of all of those proteins interacting together. So we've got the general transcription factors helping RNA polymerase bind to the promoter. We've got the activators binding to the enhancers. Upstream, we got these mediator proteins, and everything comes together, and now RNA polymerase zips down the gene to do the transcription. Okay, so okay, I think I am up and going again. So could I just have a confirmation that I'm actually up and going again? Okay. All right. There is so much to this unit. So I'm not quite sure exactly where I cut out, but here's the activators binding to the enhancer. So it's like a sandwich. You got the activators binding to the enhancer. You got the transcription factors getting RNA polymerase onto that promoter. You have mediator proteins. It all comes together, and RNA polymerase takes off and starts transcribing the gene. And so one other thing is there are repressor proteins as well, and so there might be a sequence that a particular repressor protein might bind to, and then that would prevent all of this assembly of proteins from coming together, and so now the transcription doesn't take place. Again, use the information in the prompt that is given to you to help figure out what you need to do. And you really want to think about the FRQs as not just a recall of information, but you're using the evidence and the information to figure out what they're asking you in the prompt. So read the prompt. Read the prompt. And answer the prompt. I say read and answer the prompt. RATP. Okay, so just another model that kind of shows how this all would work together. So let's say that two activators have to bind to the enhancers to get this gene to be transcribed. Remember, there's actually a lot of DNA going on here too. The gray is just representing the coding sequence. So if both activators are present inside the cell, we get transcription. If only one activator is present, maybe you get a little transcription or no transcription at all. If you have all of the activators present, but the repressor is also present, remember that repressor is going to prevent everything from coming together, and so you get no transcription. And so why are there activators in the cell? So this links back to Unit 4, where we had signal transduction pathways, cell signaling. So one scenario might be that a growth factor, so up here we got a growth factor binding to a receptor that's initiating a signal transduction pathway, and then that results in activators being now present and active inside the cell, causing transcription to take place. And once there's transcription, we get translation at the ribosome, and we get the proteins, and then those proteins cause the cell to do something that it wasn't doing before. So differential gene expression. Okay, so coordinated gene expression. So you might inside your cell have a whole set of proteins that need to be activated together to carry out a cellular response. And so here's a scenario. There's a stressor. Something happened, like there's drought, and that's going to activate some transcription factors. So we're in a plant, let's say. Those transcription factors, it's the same transcription factor that's going to bind to all three of these genes. So all three of these genes have a place, a sequence for this particular transcription factor to bind. And so all of these genes are going to be turned on. They're going to be transcribed at the same time. They're not going to all make the same protein because there's three different genes, but now you have these set of proteins that are going to carry out the stress response inside the cell. So this is what we call coordinated gene expression. And again, the essential knowledge is down in the speaker notes that you can go back to when you see the slides. So then we also have sequential gene expression during development. So just another example, and I think like heads are exploding. There is so much information here. Again, the big thing to remember is DNA codes for RNA. RNA then gets transcription. DNA is copied to RNA, transcription, and then RNA is used to assemble those amino acids at the polypeptide. Translation, we get a polypeptide, it folds, we get a protein. So this is how we go from gene to proteins and then traits. So the way this one works is we got this embryonic cell. It's supposed to, its descendants are supposed to become a muscle cell. Well, this cell gets a signal, and it's supposed to, right now all the genes are off. It gets a signal to turn on this MyoD master regulatory gene, and it makes this MyoD protein transcription factor. Again, this is a myoblast. It's going to become a muscle cell. Its descendants are going to become a muscle cell, but it's not a muscle cell yet, so it's determined. This transcription factor then binds to itself, so it increases the gene expression of itself. It also binds to a bunch of other, so then it binds to this gene. This now makes a different transcription factor that turns on other genes, and now the cell is making muscle-specific proteins. It says there like it's blocking the cell cycle. This is basically how embryonic cells differentiate into specialized cells. And there's this series of transcription factors that turn on genes at the right time in the right place, and so organs can develop during development. Now, mutations to these can actually change body plans, and that's how we can result in different characteristics in different species. Okay, one last thing here. See how bad I am. On over. Okay. So there is this thing called RNA interference, and the idea here is that there's actually some sequences in our DNA that make these little microRNAs. They're transcribed. They're never translated. They join with these protein complexes, and then based on how well they match up to an mRNA, they can either cause that mRNA to be chopped up. If it's chopped up, it doesn't get translated, or it can just sit there and now the ribosome can't pass it and it can't be translated. So this is just another step of gene regulation that can take place after transcription. But transcription is the main step where gene regulation takes place in both prokaryotes and eukaryotes. No mRNA, no protein. Haven't wasted all that time doing transcription. So here's one of the animations that I promised on transcription. It does get into some regulation of transcription in eukaryotes, and here's an animation on translation. So these are both really good at going over and making the process into a process and not just a series of steps. All right. Again, genotype, alleles you inherit, the DNA sequences you get, those genes are going to be transcribed. The mRNA is going to be translated. That's either going to make proteins or not make proteins, and what proteins you have inside your cell is going to determine your traits or your phenotype. So mutations, we are on the last topic. So last topic, we've already done the biotech, which is 6.8, so this is 6.7 on mutations. A bunch of stuff thrown in here. So causes of random mutations, and that's one thing that you want to remember about mutations is that they are random. They are random mistakes, errors, damage to the DNA that results in new DNA sequences if they happen to get passed on to offspring or onto daughter cells if we're talking about somatic cells. So one way is that you could have errors in DNA replication or the repair mechanisms that take place when the DNA is being repaired. So remember there's this G1 checkpoint, and then there's also a G2 checkpoint where the DNA is, they're looking for errors and they fix it. They have these proteins that fix it if it can happen. Sometimes it gets fixed and there's an incorrect sequence, nucleotide that's put in there. And now there's one DNA molecule has a different sequence than the other, and so when mitosis or meiosis happens, a mutation is passed on. Also, external factors such as radiation and reactive chemicals can also induce mutations. So this one happens to be UV. UV targets thymines that are two thymines that are next to each other, and it somehow causes them to bond with each other. And so when this DNA is copied by replication, two nucleotides would get skipped. That would introduce a mutation. All right, so point mutations. One type of point mutation is called a substitution mutation, and that is exactly what it means. So think about it as like a soccer game. You are being substituted for somebody else. You're going in, they're coming off. You still have 11 players on the field, but there's a different player on the field than was there before. So what a substitution mutation is going to do is it's going to change that particular codon. And if it happens to change the codon so that it's not the same, it doesn't have that redundancy, it's going to put a different amino acid in there. Now, we'll come back to that. So here's our normal protein right here. We're putting a different amino acid in. It might cause the protein to no longer fold in the proper shape, and then that protein wouldn't be able to do what it's going to do. So that would change the phenotype. Another thing that can happen is a substitution can result in a stop codon, a premature stop codon. And so now instead of continuing on and making the whole polypeptide, translation stops there because stop means stop. And now we have a protein that's not the right shape. So this is how, remember, the mutations occur in the DNA. They get passed on. The DNA then gets transcribed by the cell that's going to express that gene. But because there's a different nucleotide, it's going to change that one amino acid, or it's going to stop the polypeptide before it's supposed to stop. Now, it doesn't always cause a problem. So because of that redundancy, we call this a silent mutation. So it was a C, now it's a T, but it still codes for the same amino acid. lysine. So this would be an example of a substitution mutation that would have absolutely no impact on phenotype. It doesn't change the amino acid sequence. The protein is not changed. It still functions the way it does, even though there's a different DNA sequence. We call that a silent mutation. Nonsense mutations means it just like stops and it's nonsense. It just doesn't have the full, it can't fold properly because it doesn't have all the amino acids that it should have. And then missense, that's when a different amino acid gets put in. So this one is actually, let's go to this one. So this is let's go here. So this is a different amino acid, but it's still a basic amino acid. So you haven't really changed the chemical property of the amino acid, so that's probably not going to change how the protein folds. So you can actually have an amino acid, going the wrong way. You can actually have a different amino acid in that spot, and the protein is still folded the right way, and it still functions the right way. It hasn't changed phenotype, hasn't changed the trait. But if the amino acid that is now there because of the substitution has a different chemical property, it's going to most likely affect the folding of the protein, and it's going to cause probably a loss of function of the protein. And then the phenotype would be a result of that protein not doing its job. Whew. All right, frame shift. So this usually is when we have an insertion or a deletion of one or two nucleotides. Remember, the mutation happens back at the DNA. DNA gets passed on to offspring. Gene is now being expressed in the cell that has this altered sequence. And what's going to happen with a frame shift is, remember, the ribosome reads 3 nucleotides at a time. Codons are 3 nucleotides of the mRNA. But because you've either added or taken away 1 or 2 nucleotides, you're going to shift that reading frame over. So we call this a frame shift. It causes a shift in the ribosome's reading frame. So from that point on, all of the amino acids are going to be different in the mutated allele. Most likely, not going to fold into something that's going to do anything. And so this diagram just gives you a summary of the different point mutations. I'm telling you, there's a ton of stuff in this unit. So now, we got to talk a little bit about how mutations cause or are a source of genetic variation. And whether a mutation is detrimental, beneficial, or neutral depends on the environmental context. So what this diagram is showing us is here's the original allele. Here are the mutated variations. This variation, not beneficial. So maybe like this is a white mouse living on white sand. Oh no, that would be good. Maybe this is a white mouse living on dark sand. It's not going to survive. It's going to be selected against. So that particular mutation is not going to get passed on to offspring. We're talking natural selection here. These two, they're fine. And then reproduction and mutations occur again. So now we got this lighter one here. And maybe there's been a change in the environment, so this is no longer favorable. And so these guys don't survive and reproduce. But these individuals who have these particular alleles, they're going to pass on their traits to their offspring, and so we've got natural selection going on. Perfect example of that is the rock pocket mouse that some of you might have learned about. So rock pocket mouse, most common trait is to be light colored. Then there is this lava flow, no longer a favorable choice. Now the mutant mice with their dark colored fur, they survive, they reproduce, and they pass on that allele to their offspring. So mutations are not necessarily good or bad. They could be detrimental, beneficial, or neutral, depending on the environmental conditions that the organism lives in. So here, being a dark rock pocket mouse, not good. You're going to be seen by the hawks. Being here, a dark rock pocket mouse, awesome. Yes, it's like being a carrier of hemoglobin when they're a carrier of sickle cell anemia version of hemoglobin when you live in a malaria-prone area. We're almost there. So just a couple more things that they've thrown into Unit 6 is how bacteria can increase their genetic variation. So definitely through mutations, giving new versions of genes. But bacteria can also pick up new variation, new genetic information through these three processes. So transformation, so coming back to those plasmids. So bacteria could die, they break open, they lyse, their plasmids are now in the environment. Other bacteria can pick up those plasmids and acquire those traits. This might be one way that antibiotic resistance might occur in bacteria. They pick up a plasmid from other bacteria. Another way is we've got these bacteriophages that typically would infect a bacteria cell and kind of hijack it and kill it. But sometimes the bacteria has ways to defend it, and so instead of causing the cell to die, it actually keeps that DNA and incorporates it into its genome. And now it has new DNA that it didn't have before. And then this last one, so it's called transduction. And this last one is called conjugation, and this is where bacteria with specific genetic factors, they can actually, one cell can give another cell directly a plasmid. And so now both cells have that genetic variation. And then we have viruses that can recombine in their host cell. So we've got a human influenza virus and a bird influenza virus. They both infect the same pig cell. The viruses are assembled inside that host cell, and then what comes out is now a mix of those two viruses. And this is how those novel viruses can come about, novel viruses like coronavirus, new versions of influenza. And yet more. There is more. We are really almost done. So errors in mitosis and meiosis can result in abnormal number of chromosomes. So you can have normal should be 2 of every chromosome. In eukaryotes, sometimes there could be 1 missing, or there could be 3. That's called a trisomy. So this can happen 2 different ways. There are errors in meiosis called non-disjunction. So it could be that homologous chromosomes fail to separate. So you see here that you've got now 2 cells with 3 chromosomes and 2 cells with only 1, which in this case they should each have 2 chromosomes. Meiosis, haploid number. Over here, non-disjunction is happening with sister chromatids failing to come apart in meiosis 2. So this is going to result in gametes that then have an abnormal number, and then this could come up with what we call an aneuploidy zygote. That's going to cause sterility. And I'm not going to go into that in detail. All right. So here's one example would be trisomy 21, which causes Down syndrome, and so that can happen in humans. I'm done. So what follows are 5 FRQs from past AP exams that I pulled out that have to do with gene expression. After each one is my explanation of what the correct answer would be. Again, these are not going to necessarily be like the FRQs that are on this year's AP exam, but they are a good way to help you test your understanding of some of these basic concepts that we've gone through here. So there's 5 of them, and each one has a video explanation. And that is that. So thank you so much for hanging out with me. Maybe you couldn't make it the full amount of time and you've come back. Remember, the slides are available. There's information in the speaker notes. Those videos that I linked for replication, transcription, and translation are really, really helpful. Thank you to the moderators. I could not have done this without you. There was just way too much to get through that I just couldn't even keep track of that chat. So thanks a lot. I hope that everyone is doing well, and I'll put my email in the slides, and you can feel free to to send me a question if you have one. All right. I am going to sign off. Be well.